So I'm here to present this functional craniological perspective and the evidence this approach can bring to brain evolution. You know that we don't have brain, we have brain case. This is the main problem in paleoneurology. So everything we tell about brain is by an inference. We will never have a brain of a Neanderthal, so everything will be an inferences beginning from the shell, which is the skull. So this is strange because paleoneurologist is a neuroscientist, but who don't study the brain, but he studies the skull. And we have the endocast. This is strange because paleoneurology always worked with the endocast, but endocast does not add any information with respect to the skull. I mean, the brain was the positive, the skull is the negative mold of the brain, and the endocast is a positive mold of the negative mold of the positive brain. There's no adding of information around this chain, and you can study, paleoneurologists just study the endocast, the endocranium. We are primates. We are all our perceptive system is based on to see and to touch. So we are not happy with a cavity. We are not happy with some empty volume, and we need some objects we can touch, some objects we can handle. And this is why we largely rely on endocasts, even if this is not necessary. I begin with this sentence by Darcy Thompson. The skeleton that we see in a museum is poor and even misleading picture of mechanical efficiency. From the engineer's point of view, it is a diagram showing all the compression lines, but by no means all the tension lines of the construction. It shows all the struts, but few of the ties, and perhaps we might even say none of the principal ones. And keeping on with Darcy Thompson in another part on the book on growth and form, morphology is not only a study of material things and of the forms of material things, but as its dynamical aspect, under which we deal with the interpretation in terms of force of the operations of energy. This is very useful to understand that our final target is not that object, but the process leading to the final result, which is that object. So, I guess that these points are important to understand paleoneurology. An endocast does not add information compared with an endocranium. An endocast is not a brain, so many times it seems that many people forget this point. An endocast shouldn't be studied without its skull. An endocast is the result of the integration with that skull. If I see only the endocast, I see only a part of it, of the system. An endocast is the rough geometrical result of a dynamic interactions between soft and hard tissue. I should not be interested in the object. I should be interested in the process. I will divide this talk in this 5-point introduction functional chronology. Then I will talk about frontal areas and parietal areas, the endocranial base elements, and I will provide some conclusion suggesting using models in paleoneurology. So first of all, functional craniology, which is the main topic of this talk. We have some cells putting bones and quitting bone. This is a component in which the genetic background can be very relevant. These cells produce hard tissues, hard elements, and these hard elements physically interact with soft elements, which are the meninges, the vascular system, and the connective tensors, connective components of the endocrinium. This element have a direct contact with the brain. So this is the network interested, in which the paleoneurologist must be interested. So a paleoneurologist does not study simply the endocrinal form. After all, a paleoneurologist investigates this network, and most of all, how this network has changed in 5 million years. Recently, in the last 10 years, more and more importance has been devoted to understand not the single element, but the interaction. Finally, we accept that information of single elements may be definitely misleading. We have to understand the system, the anatomical system, with all the relationship, and so we are talking about morphological integration. So in the moment that we know we want to investigate how the skull did evolve, we have to understand the skull as a system of relationship. And it is clear, in our case, we have to add the brain. So after all, what I'm interested in is the relationship between hard tissues with soft tissues. When my hard tissues are the bones and my soft tissues are mainly the brain and the connective elements integrating bones with the brain. So, it is clear that during morphogenesis, as well as during evolution, the skull did influence the brain morphology. And at the same time, the brain morphology did influence the skull morphology. So I think that this is the really important things in paleoneurology. Paleoneurology is but the investigation on this kind of relationship. The final target should be to understand how in evolution and how much brain has influenced the evolution of skull, and skull has influenced the evolution of brain. I used to divide a kind of relationship within this kind of systems, which are functional relationship and structural relationship. The first ones are associated to functional process. So physiology, biochemistry, metabolism, the machine must provide some biological functions. The second relationships are more physical, biomechanics, morphogenesis. I mean, all these elements get in contact. So they, apart from the final physiological target, they have some forced physical interaction. So they have to reach an agreement on how to do that. It is clear that many functional relationships influence structural relationship, and that many structural relationships will influence functional relationships. And it's also clear that there's no black and white separation between these two blocks. This is a general theoretical framework. So most of the relationship I will find in the brain cage system will not be only structural or only functional. There will be some relationship which will be mainly structural and other relationships which will be mainly functional. But of course, it's not a matter of black and white. It's a grayscale separation, and it must be kept in mind that this is just a theoretical scheme. Think about this system. We have genes and we have characters. We know that In many situations, one single gene can influence so many characters. It's called pleiotropy. But at the same time, we know that a single character is probably influenced by several genes, which is polygyny. Then we have integration. Integration between genes and integration between characters. In the second case, the integration between characters are, for example, the structural relationships. And then we have the third component, the environment, influencing at the same time gene expression and character expression through phenotypic plasticity and acclimatization. So, changing the ontogenetic process or simply the physiological response of the individuals. So, taking into account this network, it is clear that, first of all, if selection acts upon one single gene or one single character, many other genes and characters will change as secondary effect. Two, the whole change will be then weighted in terms of fitness. So the change will work or not depending upon the overall change. I mean, one very bad character may be positively selected if it is associated with a super good character, or else a good character can be negatively selected if it is associated with a super bad character. Third, many changes have no effect on fitness or adaptation. When one character changes, all the other change, and many changes will not influence the number of some, which is the fitness. And fourth, some secondary changes may be reinvested in new function. Maybe there is some secondary function, secondary changes not relevant at a given time, but that may have successively a new function. It's the case of exaptation in the sense of Stephen Jay Gould, and many times we think about physical characters, but in this case, exaptation is very strong for behavior. I always do the example of the teeth. Maybe I have to chew strong food, so I will be selected if I have thick dental structure, or maybe I will evolve dental structure, thick dental structure as a secondary consequence, and in that moment I decide that I can eat strong food. So, a paleontologist can work on this using the tool of morphological integration, analyzing the correlation between traits, analyzing the formation of functional units, analyzing the evolution of modules, and using multi-rate statistics to reveal the functional and structural networks underlying the phenotypic variation. This is the biological scope. I mean, there may be many different scopes in making this investigation. But if a paleontologist wants to follow the biological, to investigate the biological reasons why, I think that this can be the real contribution that paleontology can give. So, we have the first evidence about a relationship between shape and form of variation with Darcy Thompson at the beginning of the past century, and then Huxley introduced the terms of allometry and all the questions of relative growth, and Simpson, George Caylor Simpson, began to introduce all these concepts within paleontology. And then, of course, Stephen Jay Gould gave the official beginning to the study of allometry and heterochrony. In the last 20 years, we have had the formalization of all this concept into the current theory of morphological integration and functional morphometrics, from the geometric morphometrics of Bookstein, passing through the morphological integration issues, and the final book by Lieberman, The Evolution of the Human Head, which is a synthesis of all these things. And in the middle of all this process, we have this paper officially introducing and naming functional craniology. So an approach to brain-brain case morphogenesis in which we can theoretically separate growth and development, terms usually used as synonymous, but they mean very different things. Growth means changes in size, development means changes in shape. And these two components, at least theoretically, are a result of different physical interaction. Pressures in the case of size changes, and tensions in the case of shape changes. So main element inducing pressures are organ and muscles. Main element inducing tensions are connectives, muscles and neurons. And then I have sutures and vessels, which role is largely not still understood. If you an image like this, it's definitely clear to see that the brain is strongly packed within the brain case. So here, during morphogenesis and evolution, we have so many structural integration between the box and the contents. And so again, I would stress this point. Paleoneurology is the study of the relationship between brain and skull. Of course, there will be a situation like in the vault in which the brain mainly will shape the skull, because in the vault surface will undergo will provide passive responses to the change of size and change of shape induced by the underlying brain with its growth and development. And there will be other areas in which probably will be the skull to shape the brain geometry, because these are areas in which the morphogenesis, there are much more functional and structural relationship, and the system will definitely be more complex. So I would expect that in the vault, brain pressure, the cognitive tensor, will lead the shape changes and size changes of the skull and of the endocranium, while in the endocranial based areas, function associated with posture, mandible, cranial base flexion, the presence of orbits, the fascial block, the ethmomaxillary complex, all these elements will give the final phenotypic result, not only the brain. So, bones, connectives, neurons, the cerebrospinal fluid, which is a very relevant component, and vessel. The point is to understand this network. The connective tensors are very interesting in this sense, because Fox cerebri and term cerebelli, they directly join like a biomechanical tensor all the endocranial bones. At the same time, they are the main, they mark the main pathways of the large vascular system, like all the venous sinus and the middle meningeal artery. So they directly are involved in the morphology of the vascular network. And finally, they are also deeply attached to all the inner brain elements. If we do an analysis of the corpus callosum variation, the strongest influence is exactly at the posterior areas where the tentorium cerebelli is attached. So, Fox cerebri and tentorium cerebelli are a very good element to study functional craniology, because they are the interface, the biomechanical interface between scope, vessels, and brain at the same time. So, if we see the endocranium, we can see that falx cerebri and tentorium cerebelli are attached to some major anchor points which can be investigated in paleontology. And falx cerebri exerts a longitudinal tension, while the tentorium cerebelli exerts transversal tension. What means tension? You can imagine this balloon at 500 cubic centimeter. If I blow much, I can have 1,000 cubic centimeter. And if I keep on blowing, I have 1,500 cubic centimeter. Different size, the same shape. And what if I put an adhesive band on the balloon, 500 cubic centimeter, blowing, 1,000 cubic centimeter, blowing, 1,500 cubic centimeter. Different size, different shape. Here we have two different models with the same program. The genetic program in both cases, it's simply blow, but the result is very different. In this case, I have changes in shape associated with changes in size. Constraints force redirection and different form, but the same model. If in paleontology I find the two extreme, I think that they are different animal, but they are not different animal. They are the same animal, scale at different size. And this is not only a matter of the big balloon with the falx cerebri being the big tensor. Exactly the same kind of force may be found at cellular level, neurons. There are so much theories associating the morphological organization of the circumvolution and the inner, the organization of the inner brain volume according to the role of the neurons themselves as physical tensor. A neuron is a little thing, but thousands of millions of neurons of the fibers can act like tensors, redirecting forces of growth and generating the shapes of the cortex. And this is very interesting because if I understand that the biomechanical property of the neurons can act like biomechanical tensors, I can do the example of the number of length of fibers. For example, changing number and length of fibers, I will change the biomechanical property of the fibers, or even simply changing density, resistance, or flexibility at the cellular component. I'm not a fan of the extreme reductionist approach common nowadays used in genetics. But I recognize that in this case, one single gene change can provide a minimum change in the biomechanical property of a neuron, which multiplied for millions of neurons can exert a very strong biomechanical differences. So moving from form and size, so size and shape, because all this biomechanical arrangement is the final biological target. But after all, in paleoneurology, we deal with size and shape changes. And we must take into account that we can do estimation of this biomechanical system. But after all, I can only measure size and shape. And if I have difference in size, from smaller to larger, I can see only this result, and I never know if this result is due simply to a rough enlargement, or if under the enlargement there is some change in the reconfiguration of the elements composing the object. For the cranial capacity, it's definitely clear. Every time we have an increase in cranial capacity, we automatically think that it must be associated with more neurons. But of course, we have no evidence of this. And an increase of the volume can be given by an increment of neurons, of axons, of glia and connective supporting element, or simply in water. And in paleoneurology, I cannot know what of this component is really associated with a size change. Exactly the same thing if I analyze shape change. In this case, I have a change of shape with no changes in size. And of course, in paleoneurology, I will have this evidence, and I will never know if the same change of shape is associated with a change in internal shape, change in proportions, in position of the element, or addition and deletion of elements. So I will have just the final result. So, endocrinal form changes in morphogenesis and evolution. First, investigate the extrinsic relationship between brain and brain case. Second, trying to understand the intrinsic relationship within the brain system and among the brain components. So, about the frontal areas. We know that the outer frontal bone in human evolution did show a huge variation, while the inner mid-sagittal profile in the last 500,000 years has changed so little. We are working on the bulging of the frontal squamma, and we can say that yes, modern humans have more bulging frontal squamma, independent upon the brow ridge, independent upon the orientation of the bone. Yes, modern humans have more bulging frontal squamma, and this may be related to some morphological readjustment of the prefrontal areas, but it's very subtle. The problem is that the frontal lobes are entangled in a very complex structural and functional system. The frontal lobes are housed into a box in which you have the frontal sinuses, the brow ridge, it's the face, the ethmomaxillary complex. So many different sources of influence. Enlow described how the anterior fossa directly interacts with the underlying fascial block. The recent inference of the town child for the hypostotic traits of the frontal bones may suggest that some frontal rearrangements could have occurred very early in the natural history of hominids. Anyway, things have changed in the last 2 million years, because if you see the very archaic species, Homo ergaster, Homo erectus, you have the frontal lobes in green, well behind the orbital roof in red. While in modern humans and Neanderthals, the frontal lobes are directly positioned upon the orbital roof. So, for small brain hominids, maybe the influence of the face could have been less important than in large brain hominids. In modern humans and Neanderthals, the relationship between the frontal lobes and the other-like face is definitely more relevant. And first, you have the physical contact. Frontal lobes and fascial structure compete for the space. And second, there's weight. The orbital roof is very delicate. And it's a super tiny layer of bone. And three, you have morphogenetic processes. During morphogenesis, the fascial block rotates under the narrow cranial block. So evidently in this process, most of all we humans, in which this process is extreme, of course, you will have some possible constraint at the point of contact. A recent theory by Michael Master, it's wonderful. It's a wonderful example of human evolution, functional craniology, and biomedicine. Michael Master suggests that this structural constraint, so extreme in modern humans, may be related to the myopia, the most important visual defect in our species. The orbit and the prefrontal area may compete for the same space. The orbits undergo a small deformation. There's no space enough to adjust perfectly, and so vision suffers a little bit of this problem. And the point is that if in a vertical dimension we have all this limited evidence of some relevant changes and all these constraints, in the lateral direction the situation is very different. Neanderthal modern humans, they have very relatively large frontal lobes. All the other hominids, they have very narrow frontal lobes. And this more or less has been always acknowledged, but maybe a long term. Simply larger brain, larger frontal lobes. A couple of years ago with Ralph Holloway, we measured this, and no, it's not allometry. At the same brain size, modern humans and Neanderthals, they have wider frontal lobes than smaller hominids. So it's not simply an allometric human share process. No, it's a great shift experienced by modern humans and Neanderthals. So we can interpret this in terms of constraint. In terms of, this is MRI, these are MRI section, coronal MRI section in frontal view, and the vertical variation is strongly constrained. Orbital roof, orbital volume, fascial rotation. So there are strong constraints limiting the variation of the vertical changes in the frontal lobes. At the same time, there's a large space availability on the lateral side of the anterior cranial fossa. First, apart from the orbit frontation in hominids, here you have the temporal muscle in a baboon, in modern humans, and the section is a calytrix. In primates, the temporal muscle is huge. The temporal muscle in primates is proportional to the muzzle length. So in modern humans, we have the most extreme orbit frontation. Orbits are definitely anterior. And we have the most extreme muscle reduction with a very a super reduction of the temporal structure, of the temporal muscle. So if our anterior cranial fossa is strongly constrained in this direction, there's so much space left in terms of evolution in the lateral proportions. Somebody also proposed that maybe the temporal muscle may have an active role. There's some genetic changes associating to the myosin structure of the temporal muscle between humans and apes, and some authors suggested that maybe a relaxation, it's not only a question of space, but also of tension. This hypothesis, I like very much this hypothesis because it remembers us the importance of the biomechanical context. But really this hypothesis is not very well accepted by the scientific community because there are many flaws, most of all in terms of timing of morphogenetic process. So, vertical spatial constraints, but lateral spatial availability, allometric relocation of the neural volume. We did three hypotheses. Why Neanderthal modern humans have wide frontal lobes? First, maybe neural adaptation. By chance, this point is the Burgos areas, so maybe some direct neural adaptation leads to function, language, whatever. Or neural redistribution. Simply, every time in evolution there is a brain size change, I cannot put brain mass vertically in the frontal lobes. So I put brain mass laterally. Third, excitation. Maybe we have this lateral reallocation for structural constraints, but then this decision has been reinvested in the new neural function. Parietal areas. You know that I published many things on the parietal areas. Parietal bones and parietal areas, we have not a strong match between the borderline of bone and cortical surface, but the parietal bone and the parietal areas are definitely, they have a very strong contact. And the morphogenesis of the one is strongly influenced by the morphogenesis of the other. So it's a very good chance to study paleoneurology. Ralph Holloway suggested that changes in parietal area was associated with the early evolution of hominids in Australopithecus and all the Lineaceous stuff. And then making the first geometrical analysis on parietal surface, Holloway found a huge variation of the parietal areas at level of hominids, hominoids, and the human species. And Weidereich, analyzing the Synanthropus, told that many decades ago, many people are focusing all the attention on the frontal areas, but analyzing this fossil record, it seems that the parietal areas are definitely more important. So it's nothing new, all these things in parietal areas. In rear view, the Endocastavelum erectus has the maximum width at the temporal level, and Neanderthal has the maximum width at the lower parietal areas. The modern humans has the maximum width at the upper parietal areas. So it is clear that the parietal areas have suffered some form change during evolution. But also here, nobody did measure this until me, Ralph Holloway, and Manny got making some very silly metrics to see how much this is real. And we published this in an anatomical record. The paper was rejected by American Journal of Physical Anthropology because the referee told that paleontology should not lose time in measuring something that can be seen by eyes. I disagree. Well, this is the result. Hemispheric length on parietal length. You have archaic humans and Neanderthals, Homo heidelbergensis on the lower trajectory, and you have modern humans on the upper trajectory. At the same hemispheric length, modern humans have definitely longer parietal surface. The bulging of the frontoparietal profile is something that has been widely described since ever in literature. And the most important thing is that we use geometrical models to understand this bulging is associated with what? The geometrical bulging is associated with dilation, geometrical relative expansion of the upper parietal surfaces. And more interesting, this pattern separates modern humans from all the non-modern humans. It's not a question of more or less. Here it seems a question of black or white. This pattern separates all the non-modern species from Homo sapiens, including very archaic specimens like skull, Katze, or even gibbly roots if you include the fascial component. I mean, this trait does not seem a question of degree. It seems a question of black and white. If we move from the skull to the brain, analyzing the rough, gross endocranial diameter, the same things. Archaic humans and Neanderthals, they lie on a allometric trajectory, Neanderthals are most of all a scale version of archaic humans. Modern humans lie on a different trajectory by virtue of the parietal proportions. And if we use geometric morphometrics on the endocast, the result is the same. This pattern, mostly associated with geometric parietal dilation, is first the major axis of correlation within the human genus. Second, it is a discontinuity between modern and non-modern humans. If we don't analyze covariation in all the human genus, but we just analyze the difference between modern humans and Neanderthals, which have the same cranial capacity, so the same size, the pattern is exactly the same. Now we know, thanks to Simon and Phil, that this parietal expansion, have a look at the picture of the Neanderthal modern comparison on the lower right, and have a look at the picture published by Simon. It's incredibly similar. So we know that this pattern is associated with an early postnatal period, that this pattern is absent in chimpanzee, and that this pattern is absent in Neanderthal. There's one small differences which separate Neanderthals from other non-modern humans. Neanderthals have a sort of little bulging of the lateral parietal surface. Homo burgensis doesn't have such lateral bulging. Neanderthal, they have always this marked lateral bulging of the upper parietal surface. At the same time, if we exclude modern humans and we analyze only non-modern humans, we can see that the largest the brain, the more compressed the parietal areas. This seems to be an allometric trajectory of non-modern species. The larger the brain, the more the parietal area became relatively shorter and bent. This pattern, present in all the non-modern species, is extreme in Neanderthal, of course. And at the same time, if we ever look to the skull of Neanderthal, just here, where the compression has been evidenced, we find supernumerary ossicles. Supernumerary ossicles are hypostotic traits associated with insufficient ossification and scarce integration between shape and size changes. So, again, when I found this, I tried to think about functional craniology and the falx cerebri. Maybe there are allometric constraints. The brain grows at a cube. The tension of the adhesive bands increase at a square power. So maybe it's a classical volume to surface problem. And furthermore, the frontal area are free to readjust their position. The occipital areas are free to readjust their position. The parietal areas are in the midway. They cannot adjust their position. So probably this allometric pattern lead Neanderthal to a sort of unbalance between size changes and shape changes, leading to morphogenetic stress and hypostotic traits. I'm quite proud of this chain of hypotheses, because the first person suggesting this point was Sergio Sergi of the Italian Institute of Anthropology, evidencing a morphological instability of the Neanderthal parietal occipital area. In the 90s, Giorgio Manzi of the Italian Institute of Anthropology proposed the ontogenetic stress in introducing functional craniology in the interpretation of this trait. And 10 years ago, I evidenced such allometric process within the non-modern human species. So it's a small contribution of the Italian Institute of Anthropology to the functional craniology stuff. So this bulging of the modern humans, well, it can be associated maybe to brain evolution, and by the way, give a solution to the biomechanical constraints, or maybe it's the escape from the biomechanical constraints, giving a new neurofunctional perspective. And as usually, we don't know the solution of this. I've tried to understand the parietal bulging is associated with what? If we analyze the overall neurocranial differences, was associated with parietal dilation. If we analyze only the parietal, we can find that the dilation is associated to a given point of geometrical expansion, which is more or less positioned there. There we have, first of all, the intraparietal sulcus, and then we have parasagittally, we have the intraparietal sulcus, sagittally we have the percutaneous and the medial parietal areas. Both areas are most of all involved in visuospatial integration, so a projection of the outer world into the inner world, and mental experiment, simulation, and all this stuff. And both areas have been present some human-specific neural element when compared with non-human primates. So both areas may be really associated with the parietal bulging. I studied the integration of the cortical and subcortical element in modern humans, and I found the strongest, there's no strong integration in the variation of the modern human brain, but the stronger signal of modular integration is just there, between the posterior subcortical areas and the parietal outline. And using the same configuration, we have analyzed the correlation with psychometric test cognition. There's no strong correlation of brain shape with cognition, with one exception: mental speed. Different variables of mental speed are very slightly associated with minor geometrical changes of the brain. And the geometrical changes are, well, if you cannot see this in red, but it's associated with exactly the same point. So, the same areas is associated with human evolution, with brain and morphological integration, and with some minor signal in cognition. So it's evidently an area which merits attention. It is important to say that parietal areas are not independent. Parietal areas, in terms of function, in terms of evolution, are associated with the frontal areas. Anytime in the fossil records, we have a change in the parietal areas, we have also a change in the frontal areas. Most of the theory, for example, of intelligence, nowadays don't talk about one point or one brain area or other. Most of the theory of intelligence talk about the frontal parietal system. And if you consider the connectivity between these areas, well, the frontoparietal areas are deeply interconnected with cortical channeling and re-entrance signaling. So frontal and parietal area must be interpreted as a whole system. And it is interesting that both modern humans and Neanderthals displayed frontal widening and parietal widening, sharing the same Mousterian tool, until modern humans add a parietal enlargement, and here we have a further change of the archaeological record. There are specimens like Jebel Root which are a bit different because it's probably a modern lineage specimen, but without parietal development. So this raises some question about the origin of modern human lineage and the origin of the modern human brain. Maybe they was not strictly associated. And second of all, Philip demonstrated that the parietal morphology is strongly integrated with the occipital morphology. So this relationship must be deeply considered, and it allows us to conclude the neurocranial stuff, moving to the endocranial base. We are talking about the anterior fossa, the middle fossa, and the posterior fossa. Well, the subcortical areas, there's nothing much to do. Subcortical areas are not of interest in paleoneurology because we don't have subcortical areas, and the shape is definitely associated with the cranial base angle. So we'll skip this. And I will move to the Marcus paper on the orbital and temporal areas. Marcus evidenced some strong differences in the orbitofrontal areas and in the temporal areas. As I told you, we are talking about elements which are in a structural interface between the neurocranial and the fascial blocks. And the same we can say for the temporal, for the middle cranial fossa. We know that Marcus himself demonstrated that the temporal areas variation are strongly correlated to the variation of the mandibular structure. So there's an intimate, functional, and structural relationship with non-neural factors. And of course, in my view, there we have situations in which the mandibular components may influence the brain component. The brain components may influence the mandibular components, but it is clear that in terms of structure and biomechanics, the mandible is a strong determinant in terms of physical interaction. So I guess that influence, considering the strong influence of the mandibular biomechanics, the difficulties in homology and landmarking, temporal lobes are very difficult to analyze in terms of points. The limited correspondence between the temporal lobes and middle cranial fossa. Middle cranial fossa is just one a minor box of the whole temporal areas. The complex morphogenesis and the very subtle differences we have there, I guess that yes, it is possible that brain morphology shapes skull morphology at the cranial base, but it is more probable that the skull base molded the brain form. In such a complex morphogenetic, functional, and structural scenario, it would be very difficult to evidence a primary, it means neural, it means adaptive, evolutionary brain changes here. So this is my guess. Neuroanatomical and neurofunctional evidence suggests that the involvement of the temporal and orbitofrontal areas in human brain evolution, but it is very difficult to find a clear morphological signal of such neural changes in paleoneurology, because the morphology of the anterior and middle fossa are so largely influenced by so many non-neural factors. And I would like if, at the end of my talk, you can give a couple of spots on this. So, it is interesting that they have also found a difference in the vascular system of the occipital area in Neanderthals, possibly associated with this widening extreme in Neanderthal. And this is, I think, very interesting because it can give some cues of increased functional asymmetry of the vascular system. And about the occipital areas, I can only say that there is a certain occipital projection in the Asian Homo erectus, but there's total absence of statistical evidence. It is possibly related with extreme platycephaly of Homo erectus. Possibly there are influence of the cranial base, and so I don't think that there are much paleoneurological information there. At the same time, Asian Homo erectus, they have projection of occipital lobes. The middle meningeal artery is shifted backwards. The cerebral lobes are shifted backwards. It sounds like a structural adjustment and not like a neural revolution. The Homo erectus in Homo sapiens, the cerebellum in Homo sapiens is a little bit forward. In Asian Homo erectus is backwards. but even in the case of the cerebellar fossa, I think that here, well, in Homo erectus, cerebellar lobes are more parallel. In Homo sapiens and Neanderthals, they are more convergent behind the encephalic trunk, but to be sincere, these are scarcely preserved area, very limited sample size, involved with occipital integration and endocrinal base integration. I think that here we are definitely looking at structural adjustment with no neural signal. Okay, so conclusion. Conclusion. I invite paleoneurologists to use modeling. We need model humans models. We need quantitative models. We need to remember that we make inference of cognition, just inferences. Paleoneurology must go beyond opinions and speculation. I like very much the term opinion. It's not a bad term. Opinions can be very interesting, can be very important, really. The problem is when I give my opinion, telling that it's not an opinion, and it's a scientific hypothesis. There, I don't give a favor to paleoneurology, because in opinion it's important as such. In the moment that I sell the opinion as a scientific hypothesis, I'm creating problems. We need more than humans model. Most of the endocrinal traits are not known for our species. Many of them may have a biomedical interest. Personally, I guess that it is better to investigate a character in billions of living individuals than in few fragmented and incomplete pieces of fossilized bone. This is why we need, if we are interested in fossils, we now need to work on living, because otherwise we will just have opinions and not scientific hypotheses. The necessity of quantitative models. Science needs quantitative procedures to develop and falsify hypotheses. Religion and politics, they work with possibility. Science works with probability. So to provide probabilities, I have to use a reliable, robust, simple. I must develop a quantitative model. Now I can introduce my fossil in this quantitative model and see if it fits or not in my quantitative model, providing an inference, not an opinion. So, paleoneurology deals with fossil anatomy and statistics. If you're working with paleoneurology, you're working with fossils, anatomy, and statistics, which mean paleontology, endocrine, and morphology. If the problem includes paleontology, endocrine, and morphology, it can be faced with paleoneurology. Other kind of problems cannot be faced with paleoneurology. And it is a narrow thing that paleoneurology is brain evolution. To provide inferences of brain evolution, I must use the paleoneurological information with all the rest of the biological and cognitive information. Paleoneurology is just a very minor part of this scenario. And if I move directly from paleoneurology to brain evolution, again, I'm not making a favor to our field. Most of all, paleoneurology is not on cognition. If I want to move from anatomy to cognition, I must contact non-paleoneurologists. I must work in terms of multidisciplinary proposal to provide reliable and interesting inferences in cognition. I used to say that we have to use a criterion of subtraction. Stephen Jay Gould proposed the criterion of subtraction for adaptation. I analyze allometry. If it is allometric, I cannot say that it's adaptation. Maybe it's a consequence. If it is non-allometric, it must be adaptation. So, similarly, I can hypothesize that an endocrinal shape change can have represented a primary functional evolutionary change only if I can reasonably exclude a secondary structural rearrangement. By using allometry integration and functional craniology, I must at least try to separate primary functional adaptations, in the case of paleoneurology, neural adaptation, secondary structural consequence, and possibly exaptation, which is a result of integration of the two. Otherwise, if we don't do that, paleoneurology will always be interpreted as phrenology, and the reputation of the field will be not that good. Paleoneurology is not phrenology. Paleoneurology should be an integration of the anatomical and functional and structural signal to understand the brain versus brain relationship and their evolution. Molecular fields are performing. Molecular fields are young fields, we can say. They are providing a super reductionist approach, like morphology did 1 century ago. I think that paleoneurology is now, and morphology in general, is sufficient aged to learn from past error and move from extreme reductionist approach using by other discipline. I think that the times are right. We have the tool to analyze integration and to analyze this functional system, and it should be a pity if we don't use this opportunity. Thank you very much.